So I'd like to start off first by talking about a few examples. Um, Here's an example where they were comparing a drug called warfarin, and they were looking at the same drug under different types of dosing algorithms or dosing system. So this was a randomized trial of doing a one-step warfarin dosing algorithm against the widely used computerized dosing system that was already accepted as the standard. So it is not just a new drug, it is the same drug, but in a different dosing system. And what they were studying, if was the computerized system was non-inferior to the existing algorithm for the primary outcome, and this was time in therapeutic range. One of the things that they said is that we have to establish a non-inferiority margin of 4.5%. So I will be talking about where do these margins come from, for example. In fact, they were able to show that not only was the algorithm not inferior, but they also were able to show that or test for superiority. So I will also be talking about how one can use these sorts of designs to actually even go as far as testing for superiority. Now, here's another example. And this is a test of implant without testing versus the standard ICD implant process when with testing. And this is the cardioverter defibrillator implantation. This was a study that was looking at non-inferiority in the hazard ratio as of 1.5 as the margin for a composite outcome, okay? And again, where does this margin of 1.5 come from? They ended up interpreting in their results that routine defibrillation testing at the time of the implant is generally well tolerated, but does not improve shock efficacy or reduce arrhythmic death. So interpretation was that the standard does not improve shock therapy or reduce arrhythmic death. This actually shows something that they should not be concluding, because you're not proving the standard, right? So you're showing that you're comparing the new thing against the standard. So even people that publish in the Lancet are having difficulties in interpreting the results of non-inferiority trials. So I'm going to be focusing on how do we interpret non-inferiority trials. My third example before I start is a pilot study evaluating the non-inferiority of a single preoperative dose versus prolonged prophylaxis and superiority of this vancomycin is not only used in regular, and their sample size was driven by superiority on feasibility outcomes. So non-inferiority trials are being used widely. What I'm going to be reviewing in the first part of this workshop is the non-inferiority hypothesis, how we go about defining it, how we select the non-inferiority margins, and then some other issues in non-inferiority trials and how to present the results. So let's start off with something that we know very well, and that is hypotheses in randomized controlled trials, where we're trying to compare the effect of one treatment, let's call it A, to that of another treatment B on a certain endpoint. And we're very familiar with the hypothesis of superiority, as I said at the beginning, because usually you want to show that, let's say, A is better than B, or A is worse than B, A is better than B, or different from B. Okay? And it could be depending on what is the outcome, you may be looking at it this way or going the other one side. Usually we do superiority two-sided. But we may also be interested in what used to be called equivalence trials, and that is showing that the difference between A and B is bounded by some value gamma. Okay. Today, I'm going to be focusing on non-inferiority. And if, let's say, A is the test treatment or new treatment, and B is the standard treatment, what we'll be looking at is it really depends on one-sided hypothesis alternative. These are usually we do two-sided for superiority, and here we do it. This is essentially because of you taken the absolute value. We are also two-sided for equivalence. But for non-inferiority, we're usually focusing on a one-sided hypothesis. So if something is bad, then if this is the standard and you're taking the difference of the new from the standard, you would like that maybe you would tolerate it to be slightly worse, but you would not tolerate it to be too worse. So the difference of this cannot, you don't want it to be too small, okay? And the opposite happens then for good outcomes. These capital delta are called the margins, and we're going to go now specifically in detail to how do we interpret these non-inferiority hypotheses and also how we select the margins. So first of all, why do we do non-inferiority margins or trials? It's because we want to show that something is no worse than some existing standard by some amount capital delta. This happens frequently when we have a regulatory approval of a new investigational treatment, usually a drug, by showing that it's not inferior to something that is known to be efficacious. So it's usually not against placebo, it's against the standard that we know to be efficacious. Or, for example, it's used quite a bit in generics, for example, in that sense. Now, another use for it is that we may want to show that one approved drug is no less efficacious than another approved drug, either for new dosing regimen, like I gave you an example, or because for some types of diseases, we want to have multiple alternatives for the patient. For example, if we have for hypertension, for high blood pressure, we may want to have different types of medications because some people may tolerate one better than the other, okay? And so we want to say, we don't need to show that it's better. We just need to show that it's not too worse than the one that's already considered to be the standard, okay? So that's why we do non-inferiority trials. Now, before I go in detail on the non-inferiority trials, I'd like to present the other hypothesis, the superiority one and the equivalence one, in terms of confidence intervals, because when we are studying non-inferiority trials, it makes more sense to look at the confidence intervals and give a confidence interval interpretation to our test of the hypothesis. So starting off with the superiority hypothesis that we all know very well, and let's assume the outcome is continuous. Usually we're comparing means. So the null hypothesis is no difference of means. Usually we do the two-sided hypothesis as an alternative. And we designed the study to detect the smallest clinically meaningful difference, little delta, and usually at the 5% level of significance with a particular power, usually 80 or 90%. And then the significance or not in a two-sided hypothesis, we know is equivalent to having a 95% confidence interval, and that difference in means would not include or include the value of 0. So for example, if we had the confidence interval that we find does not include 0, we'd say, oh, there's a significant difference between the two. And if it includes 0, we'd say there's not a significant difference. Now, we may have planned the study for detecting a smallest clinically meaningful difference, delta, but we actually test against 0. We don't test against delta. Very interesting, right? Now, if the outcome is binary or it's a time to an event, we're usually comparing risks. So we talk about the relative risk or ratio of risk ratio. And so then the null hypothesis is 1 against the two-sided hypothesis that the risk ratio is different from 1. Or if we're doing a time to event analysis, we're comparing the hazards over time, and maybe we'll assume a proportional hazards commonly done, and then we compare the ratio of hazards, ignoring time, and again, we'll have the null hypothesis of the hazard ratio equal to 1, and the two-sided hypothesis that it's not. So again, we would do something very similar. We would design the study to detect the smallest clinically meaningful ratio. Now it's a ratio delta at the 0.5 level and with a given power. And so we have the same situation, same typical graph, except that now the reference is against 1. And we're comparing whether the interval includes 1 or not. Again, we don't test against delta, we test against 1. And this is the standard superiority hypothesis that we all know. And so we look at it as the two-sided superiority hypothesis is completely equivalent to looking at whether the 95% confidence interval includes the null hypothesis value or not. And I assume everybody is fine with that. If we move to the equivalence hypothesis, now we want to show that the effects of two treatments on a certain endpoint differ by no more than some amount gamma, right? And so if it's for differences, we want that the absolute value of the difference of the means is less than gamma. If it's for ratios, now we have to say that the relative risk or hazard ratio, depending on what you're using, is between gamma and 1 over gamma. We're talking about ratios. So when do we use equivalence hypothesis? It used to be done for clinical outcomes, but it is rarely used now for clinical outcomes. It's used primarily for what are called bioequivalence studies, where you have different formulations of the same active drug compound, and they're trying to show that they have equivalent pharmacokinetics. And so this is usually for approval of generic drugs. Or for example, when they're doing vaccine manufacturing processes, they're looking to lot-to-lot consistency and basically show an equivalent immunogenicity of the first few manufactured lots of vaccine. And this is required for vaccine approval. It used to be used for what was called clinical equivalence, which is then comparing 2 or more approved treatments for an outcome to see if they are similar, okay? However, this is no longer what is used nowadays. Nowadays, we prefer to show non-inferiority because of the one-sided hypothesis being the one of interest. We're not interested so much in the two-sided hypothesis. We're interested in that they're not that similar, but that it's not worse than the other one, okay? So nowadays, so for equivalence hypothesis, for differences, you would have these very narrow margins, gamma against 0, and you want to have equivalence, you have to have the confidence interval exactly between the margins. And if it went, even if it was a little bit better, for example, but it went above it, then they're not equivalent. And if it was for ratios, now it's again, it's between gamma and 1 over gamma, and you have to be for equivalence, you'd have to be within those margins. And if you went the other way, even a little bit, you'd say they're not equivalent, okay? And so here is a study that actually some of our colleagues had worked on, and this is a trial comparing stent versus surgery for asymptomatic carotid stenosis. And they were comparing these things on embolic protection and carotid endarterectomy in very old patients, okay, cited 95% confidence interval, okay, and that was their pre-specified non-inferiority margin was 3%. So I'm going to be talking about how we come up with the margins, but let's look at the non-inferiority hypothesis. So the non-inferiority hypothesis is now called sometimes the one-sided clinical equivalence. So remember that the regular equivalence required that you be very narrowly within a gap, but if you even went a little bit better, it was considered not equivalent. And because of that, now they're interested in, okay, I just don't want it to go beyond one of the limits, but it's okay if it goes against the other one because it may even be superior. So the differences between the equivalence and the non-inferiority is that the non-inferiority is one-sided, okay? Basically, we're interested that it's no worse than the reference treatment. And if non-inferiority is supported, maybe we can subsequently show superiority. Okay? So we usually typically use the one-sided 95.75% confidence interval because that's the limits would be the same as the 95% confidence interval, but we're only interested on one side. Some people will use the one-sided 95%, and that is allowed if you want to, but many times regulatory agencies prefer the 97.5%. So if having large values is harmful, then the event rate of the test treatment for a bad thing, such as death, for example, you don't want it to be worse. It can be a little bit worse, but not too much worse than some percent of the reference treatment. And so we usually focus on the upper limit of the confidence interval. If large values are beneficial, so you want to have a good thing, then you want it to be be larger, but you'll tolerate it being a little bit worse, but not too much. So then you focus on the lower limit of the confidence interval. And that's the only thing that is a little bit tricky to follow, okay? So that's why we look at it through confidence intervals, okay? So let's take the last thing. So something is good, then you would like it to be better than the reference. So let's say you're looking at differences, and so you're going to be compared against zero. If your confidence interval is above zero, it's superior, right? But if it is, let's say, not superior, right? It can be worse because it's less than zero. But if you have your non-inferiority margin as minus delta here, and you've specified this, but the lower bound of the 95% confidence interval does not go beyond minus delta, then you say you're not inferior. If it went below that, you'd say it's inferior. If you're looking at ratios, it's very similar diagram, except that now we're, of course, comparing against one, right? So here's one is over here. But again, you would focus on the lower bound of the 95% confidence interval. If it's a bad outcome, it's the exact opposite, right? So if you're taking the difference between the new versus the standard, then it would be superior if it is below zero. The entire confidence interval is that. But it would be non-inferior if it doesn't go, the upper limit of the upper bound of the confidence interval doesn't go beyond delta, plus delta, okay? And so you're focusing on this side of the difference. If it goes beyond that, it is then considered to be inferior. And similarly for ratios, this would happen. And so this is how we go about doing non-inferiority trials. What we have to do is specify this delta. So this non-inferiority margin is the crucial thing when you are planning and thinking about the non-inferiority trials. And are the margins related? Remember that delta is the margin for a superiority trial. Gamma was the margin for an equivalence trial, and delta is the margin for a non-inferiority trial. And yes, logically, these margins are related, and they're related. So for example, if I'm looking at differences, to show something is equivalent, they have to be very, very tightly close to each other. So therefore, gamma is usually very small. For non-inferiority, because you're doing one-sided, usually you'll tolerate something slightly bigger than gamma. And so you'd be, in this case, I'm looking at the other side, so I'm looking at I didn't want to crowd that other side, but you're looking at um, minus delta, or in this case, I could have put delta over here. Sorry, I could have put, if there's a mistake here, I should have put delta on this side. So delta can be a little bit bigger than gamma. Now, usually to show superiority, you wanted to show that it is the smallest clinically meaningful difference for being superior. So delta is usually a lot bigger, and therefore it's a lot bigger than capital delta. Okay. Now, the important way of thinking about these margins is that for showing superiority, this little delta is the smallest clinically meaningful difference for being superior. For non-inferiority, we want the largest clinically tolerable difference to claim not inferior. And to show equivalence, we want the largest clinically tolerable difference to claim that they're similar to each other. So I've highlighted in red that these are clinically meaningful or clinically tolerable. So it's not a statistical decision of these margins. It's the contextual clinician investigator that determines this. However, for non-inferiority margins, regulatory agencies have come about and said, we don't let you decide what is tolerable. We will decide what is tolerable. Okay. So some common pitfalls to avoid is that if you're testing for equivalence and you can conclude significant equivalence, this does not necessarily mean that if you had tested for superiority, you would not have found one. So just because you show equivalence doesn't mean that you would not have shown that there is a difference. And if you test for superiority and do not find one, this does not necessarily mean that you can conclude equivalence. This way, it's like concluding the null hypothesis, right? So we cannot do that, even if the study power was high. And of course, we always have to remember that statistical significance has nothing to do with the importance of the result, okay? So that's why clinically important margins are determined separately from statistical significance considerations, okay? So how do we select the equivalence in non-inferiority margins? First thing to remember is that these things must be selected a priori. You cannot do that once you start the study. And if you're going to do this for some medication that is going to be then reviewed by a regulatory authority, they must approve your choice ahead of time. And it's a clinical decision. It's not a statistical decision. Okay. Now, historically, the margins were set by clinically relevant difference for each indication. And so for continuous parameters, it was the some percent of the expected standard mean, or some percent of the difference between standard and placebo used to be. And for binary outcomes, it was the difference in proportions in an additive scale or ratios in a multiplicative scale. So in a multiplicative scale for good outcomes, it was 0.8, and for bad outcomes, 1.25, because they're the same, because there's the reciprocal of one or the other. Who established this? This was the US Food and Drug Administration. And this is what everybody in the drug companies wanted to get their drugs approved in the United States. So therefore, that's what everybody used to use. Okay. Nowadays, there is a more detailed guidance from the US Food and Drug Administration that people are adhering to. And so the new guidelines were established in 2016. You can download this from the internet. There's the non-inferiority clinical trials to establish effectiveness guidance for industries by the US Food and Drug Administration. So they came out in 2010, but then governments takes a long time to get things done. And so they were approved in 2016, okay? What does that do? It says you have to go through a process to determine the margin. So you first have to find what is M1, and M1 is the effect that the standard treatment has, or the control treatment has. what it would be reliably expected to have. Then you find what they call M2, which is the clinical margin. And the clinical margin is the largest effect of the test compared to the reference that would be clinically acceptable. Another way of interpreting this is the smallest difference which the patients perceive as beneficial and which would mandate, in the absence of any problems with side effects and excessive cost, a change in treatment. So it's the minimal clinically important difference that you want to preserve. So M2 is usually less than M1. Okay? Let's see how this works.